Hello friends, today we are going to study about protein denaturation. So first of all, what do we mean by protein denaturation? As we all know that the protein 3D structure depends upon its primary structure. The 3D structure is stable, but it is moderately stable. What does it mean? It means that it will remain stable if the surrounding condition is totally favorable. If the conditions surrounding will change, we can observe change in its 3D structure. Now, the change that leads to the loss of function is known as denaturation. Mostly, the denatured protein exists in a set of partially folded state, which means that if we put any protein in a denaturing conditions, it is not like that it will just open up and convert into a straight chain polypeptide. Mostly, it will remain folded only, but not completely partially folded. Now, what are the denaturants that can affect the structure and eventually the function of a protein? Now, first factor is heating. What happens when we increase the temperature? Let's see one graph. What we can see, when the temperature is increasing up to a particular point, there is no effect in the protein structure. The structure remains stable. Then further increase in temperature will denature the protein. Such curves tells us that protein denaturation is a cooperative process. What do we mean by cooperative process? That means the instability in one subunit will pass the message and make the another subunit unstable. Again, the instability of the second subunit will make the third unit unstable. So such pattern is known as cooperative pattern or cooperative process. What kind of bond heating will disrupt? Mainly the hydrogen bond. Other denaturants include pH. What will happen if we change the pH? pH affects the charges or the ionization states. So altering the pH will disrupt the electrostatic interaction. What will happen if we add organic solvent, urea and detergents? They mainly associate with nonpolar residues. Where we can find a nonpolar residue in a globular protein? We already studied in the previous video. If this is a globular protein, we'll find the nonpolar residues in the interior of the globular protein, making a hydrophobic core. So that means organic solvents, urea and detergent disrupts the hydrophobic interaction and hence disrupts the core of the globular protein. There are other ions and small molecules that all increase the solubility of nonpolar substances in water. For example, guanidinium ion and urea. If the nonpolar substance becomes soluble in water, again the core will get disrupted. So these are few denaturants that can denature protein. But as the protein tertiary structure depends upon its primary structure, therefore, as soon as the denaturants are removed from its environment, it will again fold in its native conformation. Let's first understand the renaturation of the protein. First of all, this RNase A was used to study the renaturation process of a protein. Now, say for example, this is some protein A. This is the native conformation of the protein. These blue color bonds are the disulfide bonds. That means how many cysteine residues this protein have? Here one cysteine, here one cysteine, here one cysteine, here one cysteine, cysteine. So how many in total? How many bonds? 1, 2, 3 and 4. So number of cysteine molecules? 8. So this is the native conformation of a protein having 8 cysteine molecules and hence stabilized by 4 disulfide bonds. Now, if we add denaturants like urea and mercaptoethanol. Mercaptoethanol is a reductant. Urea is a what it disrupts? Hydrophobic core. So what will happen to this protein? The protein denatured. It becomes unfolded. Correct? All the disulfide bond it got reduced, and hence now all the cysteines are free, so no disulfide bond. Because urea is added, that means hydrophobic core also got disrupted. So now the protein is totally in an unfolded conformation. Now, suppose in an oxidized environment, we removed urea and mercaptoethanol. We found that protein got folded exactly in its native conformation. In an oxidized environment, when we removed the mercaptoethanol, that is a reductant, and urea, then we found that protein got folded exactly the way it was earlier, before the use of these denaturants. What else we can do? Let's remove the mercaptoethanol, leave the urea as it is. So that means now in the denaturation condition, the urea is still present, but we removed the mercaptoethanol in the oxidized condition. So what we will find is the protein will fold, but the way it will fold will be different from its native conformation, and hence this protein will become non-functional. Why it happened and what it suggests then? We removed mercaptoethanol, that means disulfide bond formed, but we does not remove the urea because urea disrupts its hydrophobic core. Hydrophobic core is stabilized by hydrophobic bond, which is a kind of weak interaction. So that means for proper folding, disulfide bond is a covalent bond. So for the proper folding and the proper positioning of disulfide bond, these weak interactions are also very important. And that means we can say that the protein 3D structure depends upon its primary structure. Why so? Because the primary structure contains amino acid sequence which is responsible for forming the core or the exterior. The sequence has not disturbed due to denaturants. Only the 3D structure has got disturbed. So once we will remove all the denaturants, then because the message is already stored in the primary structure, the protein will be reformed in its native conformation. And removing only the reductant proves that weak interactions are important for proper folding of a protein, for proper placement of disulfide bond. So this experiment proved that weak interactions are important and also that the primary structure is responsible for the 3D structure and eventually the function of a protein. Now, protein can fold spontaneously into their native conformation under physiological condition. This implies that a protein's primary structure dictates its 3D structure. Now, proteins are dynamic. What does it mean? That means the protein structure is not very fixed or rigid. Flexibility is allowed. Where the flexibility can be allowed? Suppose this is a protein in a folded form. The amino acids are present like this. So the amino acids whose side chains are extended outwards, because solutions are running from its surrounding, these side chains are prone to disruption in already formed bonds between them. Because of this wave, what happens is sometimes if they are bonded like this, then due to this wave, what happens? Sometimes this bond will break and some new bond with other side chains will form. So this much flexibility is allowed mainly to the amino acid side chains that are extended outwards. And this phenomenon is known as conformational flexibility or breathing. Now, intrinsically disordered proteins. Intrinsically disordered proteins means there are some proteins that remain in a disordered form. They become ordered when they have to perform their function. So such proteins, they lack defined structure, and such proteins are mainly rich in certain polar and charged amino acids as mentioned here. They generally lack the bulky hydrophobic groups. 33% of eukaryotic proteins are intrinsically disordered protein. For example, CREB, cyclic AMP response element binding protein. This protein remain in a disordered form when it is free. They only get folded when they have to interact and perform their function. So such proteins are called intrinsically disordered protein, which remain in a disordered state and convert into ordered state when they have to perform their function. So that's all for protein denaturation. Now one question from my end for you all, that is, would intrinsically disordered polypeptide segments contain relatively more hydrophilic or hydrophobic residues? Explain. Do let me know in the comment section. If you find the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.